So today we're very pleased to have with us both Caroline Miranda and Haiqiang Dou coming to us from the University of Gothenburg, Sweden. They are going to be talking a little bit about gap junction coupling and islet delta cell function in health and disease. Just a little backstory, a little background. The pancreatic islets contain beta cells and alpha cells, which are responsible for secreting two principal glucoregulatory hormones, insulin and glucagon, respectively. However, they also contain delta cells, a relatively sparse cell type that secretes somatostatin. These cells have a complex morphology, allowing them to establish an extensive communication network throughout the islet despite their scarcity. Delta cells are electrically excitable cells, and serotonin is released in a glucose and K-ATP-dependent manner. Somatostatin hyperpolarizes the alpha cell membrane and suppresses exocytosis. In this way, islet somatostatin potentially inhibits glucagon release. Recent studies investigating the activity of delta cells have revealed they are electrically coupled to beta cells via gap junction, suggesting that the delta cell is more than just a paracrine inhibitor. In this review, they summarize delta cell morphology, function, and the role of somatostatin signaling for regulating islet hormonal output. A distinguishing feature of this review is that they attempt to use the discovery of the gap junction pathway together with what is already known about delta cells to reframe the role of these cells in both health and disease. In particular, we argue that the discovery of gap junction communication between delta cells and beta cells provides new insight into the contribution of delta cells to the islet hormonal defects observed in both type 1 and type 2 diabetes. And this reappraisal of the delta cell is important as it may offer novel insights into how the physiology of the cell can be utilized to restore islet function in diabetes. Welcome to both of you, Caroline and Haiqiang. And if you'd like to sort of take it away, you can start to share slides.
Okay. Well,
actually, let me just reframe this. I did introduce you guys as both at University of Gothenburg, but I see that I was mistaken that Caroline is at University of Oxford.
Yes, well, currently we're both in Gothenburg at this present moment, but I am affiliated to both Oxford and Gothenburg, whereas Haiqiang, he is affiliated to Gothenburg.
Okay, great. Thanks for clarifying.
Yes. Well, thank you all for being here. Thank you for this opportunity of sharing a little bit of our work with you. So today we're going to talk a little bit about the disturbance of cell-to-cell crosstalk in type 1 diabetes. So just a brief introduction. Embedded in the exocrine tissue of the pancreas are the Islets of Langerhans. And there's these microorgans that are made up of the cells that together orchestrate glucose homeostasis. And the main cells that we can see in the islets are the insulin-secreting beta cells, the glucagon-secreting alpha cells, and the less common type, the somatostatin-secreting delta cells. Whereas insulin and glucagon are glucoregulatory hormones, the role of somatostatin is a little less known than the two previous hormones. And here we have a diagram of the secretion of these hormones in relating to the glucose concentration in the blood. So when glucagon is high at low glucose, and as we get closer to the normal glycemic concentrations of our 5 to 7 range millimoles per liter, then we see the glucagon reaches its minimum concentration, whereas insulin is low at low glucose and increases as glucose concentration increases. The same trend is seen for somatostatin. Glucose regulates islet hormone secretion. When we eat a meal, we're going to experience an increase in blood glucose. This is going to be sensed by the beta cells of the islets of Langerhans, who will secrete insulin. Insulin is going to signalize all the other cells in the body to take up this glucose, removing it from the bloodstream and restoring the state of normal glycemia. Can you see my pointer here, by the way? Do you see my mouse?
Yes, I do. I see it perfectly.
Restoring the normal glycemic state. When we exercise or when we fast, then we're going to experience the lower blood glucose. And this is going to be sensed by the alpha cells, who will then secrete glucagon, that stimulating gluconeogenesis and glycogenolysis will put some of that stored glucose back into the blood, once again restoring normal glycemia. This last part, called hypoglycemia-induced glucagon release, is a life-saving mechanism, because our brain would die within minutes without glucose. And in fact, a complication that is experienced by patients with diabetes type 1, but even in some cases type 2, called a iatrogenic hypoglycemia, is when this mechanism is faulty, and because of the treatment with insulin, glucagon is unable to be secreted in the appropriate manners, will not be able to restore the normal glycemia. And this is a potentially fatal complication, by the way. So delta cells, they are small in number within islets, 5% in mouse and about 10% in human. The cytoarchitecture of the islets, they also are differ from mouse to human. And here we see a mouse islet in a cross section where the beta cell mass is clumped in the middle, forming the core. And the delta cells and the alpha cells, they form this 3D network around the beta cell mass, making the mantle of the islet. In human, what we see here in this cross section is these different cell types is scattered across the islet. And here we have a 3D model of a mouse islet as well, showing this a 3D network that the delta cells form with the alpha cells, as you see here, the delta cells in green and the alpha cells in red. And this empty space here in the middle is supposed to be the unstained beta cell mass in the middle. And this is also in mouse, by the way. They have these processes that are meant to make physical contact with different cell types in the islets. So they have these very long processes. They are longer than the cell body. Here we have an illustration of a delta cell. And here we have a real picture, a microscope picture of an alpha cell, of a delta cell. And this delta cell is genetically encoded with a fluorescent tag that you can see in green, and it has been stained by somatostatin that you can see in red. And as you see here in the ends of the process of the projections, somatostatin is present in the entire cell, including the ends of the projections.
And can I just ask you quickly, Caroline, is that image on the bottom right, is that delta cell kind of embracing another cell? Oh, it almost looks like it's the processes are kind of like surrounding another cell.
It could very well be. In this particular cell, what we have is only delta cells. Sorry, in this particular islet, what we have is only delta cells. So it could very well be embracing another cell, absolutely. But well, unfortunately, in this, we can't say anything. We can see that for sure, because we only have staining in the delta cells.
Okay, and can I ask, do you see a change in these processes, in their length, in the shape of the processes?
Pretty much, you see delta cells of many shapes and sizes. The processes, they are usually longer than the cell body, but that's not really a rule either. But you do see islets of all shapes and sizes, and you also see these delta cells. They differ, but they do have certain aspects. For example, the processes and somatostatin granules in the process is common to the pretty much all the cells.
Thank you. Interesting.
What you see here in this model is a 3D model showing that the delta cells, the processes that you see here, they reach towards the core of the cell. So these processes, they do not only reach in the X and Y manner, but also in a Z manner to make contact with islet cells that are inside the islet. And the islet cells, they communicate in a paracrine manner as by the secreted effectors here as we see, and in an electrical manner. Beta cells, they communicate with each other via gap junction, and now it's becoming more and more evident that beta cells and non-beta cells do communicate with each other via gap junctions as well. Delta cell and beta cell coupling. So somatostatin is a potent glucagon inhibitor. In our studies, we have seen that about 40% of alpha cells, they are under tonic inhibition by somatostatin. And this is in low glucose, when glucagon secretion is supposed to be at its highest, and it's also in the islets of healthy mice. So mind you, when these cells are supposed to be secreting most the glucagon, about almost half of these cells, they are being constantly inhibited by somatostatin. However, when we test static batch incubations and we look at insulin, this is not the case for insulin. So insulin remains unchanged, as you can see here, by the application, the presence or the absence of somatostatin. So the reader can end up wondering if the function of the delta cell is merely to be a paracrine inhibitor to alpha cell. So to answer some of our questions that we have developed some tools based on optogenetics. In optogenetics, we use light-sensitive proteins that we put in mouse cells, and we are able to control biological processes just by shining light. So this particular protein here called chenorodopsin from an algae, we have put this on beta cells of mice. Well, not and this protein is also tagged with a fluorescent tag. Sorry. So when we put this protein in the beta cells of mice and we isolated these islets and we shine light on these islets, we can see here at a glucose concentration where beta cells would normally be inactive, by shining light on these islets, we see that we are able to elicit electrical activity from these islets, from these cells, from these beta cells. And when we look at the insulin secretion by these islets, we also see that insulin increases about a threefold in a lights on paradigm compared to the lights off, showing that this is a true activation of these beta cells. So we were able to activate the beta cells, and when we looked at the delta cells from these islets where the beta cells were being activated by light, we saw that when we shine light on the beta cells, we were able to observe electrical activity in the delta cells. And lo and behold, when we looked at the secretion of somatostatin from these islets, we also observed a massive increase on somatostatin that can only be explained by electrical activation of the beta cells. Moreover, when we tested to apply inactive current to delta cells, we see that they are susceptible to inactivate to hyperpolarization. So we apply the negative current to these delta cells, and they do become hyperpolarized, showing that they are susceptible to inactivation. And with some previous gene expression studies, we also see that delta cells, they are rich in this type of gap junction. This gene that codes for connexin 36, it's abundantly present in delta cells. So collectively, we are able to devise sort of a model where we propose that the delta cells, they're normally clamped by the beta cell that keeps injecting this clamping current via gap junctions to the delta cells. In type 1 diabetes, what happens is that due to the death of the beta cell mass, this connection is severed, and delta cells, they are spontaneously active then. Indeed, when we, in the lab, if we take islets and we disperse them, then the delta cell, they're normally inactive, they will become spontaneously active. We have observed that. So to summarize this part of the talk, the delta cell has a much more complex role than previously thought, dynamically regulating the output of both the glucoregulatory hormones, being that the paracrine inhibition of glucagon in alpha cells and coupling to beta cells via gap junctions. So I'm going to leave the floor or the mouse to my colleague Haiqiang now, who will keep talking a little bit more.
So Caroline just talked about delta cell and the beta cell crosstalk in islets in health and the type 1 diabetes. So I will move on to the crosstalk between delta and alpha cells, also in health and in type 1 diabetes. And we are also going to use optogenetics. Here we expressed the channel rhodopsin in delta cells, and we assessed the expression with the immunostaining of somatostatin and channel rhodopsin. This showed very good colocalization, and we can see the expression of channel rhodopsin on the plasma membrane. And when we shed light, blue light, to the delta cells, we can see the depolarization of delta cell membrane during light. And we measured calcium signals in both delta cells, as was shown here in yellow, and non-delta cells, the red one. And we can see that light stimulations increased calcium in delta cells, but not in non-delta cells, which we believe was a beta cell. And also light stimulation activates delta cells and stimulated somatostatin release at both low and high glucose. So the interesting thing is that how delta cell activation and how somatostatin release regulates the activity of alpha cells. We designed an experiment like this. We record an alpha cell with patch clamp, and then we stimulate the delta cell clusters with blue light. And here, if we recorded a cell, an alpha cell, which is close to delta cell clusters, we can see that the membrane potential was repolarized very quickly during light stimulation of delta cell clusters. However, when we choose an alpha cell which is far away from delta cell clusters, the very short light stimulation did not have any effect to electrical activities of this alpha cell. But when we prolong this stimulation, the alpha cell was eventually hyperpolarized. So it shows that delta cell regulates alpha cell activity in a distance-dependent manner. And also, we measured exocytosis from alpha cells during photoactivation of daughter cells. It's very dramatic that during daughter cell activation and somatostatin release, alpha cell exocytosis was suppressed and recovered after withdrawal of light. And for glucagon secretion, light activation to daughter cells decreased glucagon secretion at just low glucose concentration, but it does not have any effect at 6 and 20 millimole glucose. So what happens to delta and alpha cell crosstalk in type 1 diabetes? As Caroline said, the somatostatin release is increased a lot in type 1 diabetes. And for the glucagon secretion, it's decreased a lot compared to the normal conditions. And we did this experiments in the NOD mice, which is a type 1 diabetes model. And also in islets from type 1 diabetes donors, we saw the similar stuff that the somatostatin release is increased in type 1 diabetes. And the glucagon secretion is not regulated anymore by the glucose concentrations. However, when we add the somatostatin receptor antagonist, it increased the glucagon secretion, which means that the alpha cells, they are on inhibition by somatostatin in type 1 diabetes. So this is our general idea about the crosstalk between islet cells in health and in type 1 diabetes, that at normal conditions or in healthy people, that daughter cell somatostatin release only affects the neighboring alpha cells, and most daughter cells are surrounded by beta cells because of the large number of beta cells. And in the alpha cells, which are far away from daughter cells, they are not affected by somatostatin release. However, in type 1 diabetes, the daughter cell is becoming spontaneously active and release more somatostatin. And also the alpha cells, they are getting closer to delta cells because beta cells are gone. So these alpha cells, they are becoming silent and they can't release glucagon anymore. This is the reason of low glucagon secretion in type 1 diabetes models. And to treat this, we might apply tebutamide, or the somatostatin receptor antagonist, which we can restore the glucagon secretion in type 1 diabetes. And we would thank Patrick Grossman, who is the PI of the labs both in Gothenburg and Oxford. And because some imaging data is provided by Johan, and the staining data provided by Ray and Chen in Oxford. Thank you.
Thank you very much. That was really fascinating work. Just amazing. And I'd like to open it up to some questions. We have some people in the audience. You can either unmute yourself or raise your hand, or you can also put something in the chat if you like. I like this slide here that you guys went back to. So you're proposing this SSTR2 antagonist as a treatment. Has that ever been tried historically in T1D patients?
No idea. I don't think so. I don't think there are any trials yet in T1D.
Sure, no, I don't know. Not strong knowledge.
Yeah, so that looks like a very interesting clinical trial, right? To sort of titrate the the remaining secretion of or modulate the secretion of the delta cells, correct?
Right, that's the idea, to rescue whatever alpha cells and get them uninhibited, if you will.
Yeah,
Yeah, to get them restore the ability to release glucagon, and
which would be very helpful in a
hypoglycemia.
Yeah, in a hypo episode, right? Because that could be, if they could kick in and come online, the lows might be less low. It's really fascinating. Amir, would you like to ask a question?
Hi, thank you for this presentation. And thank you, Haik and Caroline, for this nice presentation. Just I have a question about how with optogenetic stimulate, because I'm not expert in this area. I'm just curious about how the light can stimulate the cells. I missed that point. Sorry. If you can explain in short, I will be appreciate. Thank you.
No problem. So the channel rhodopsin is a channel which is sensitive to light, and it's a cation channel. So when we shed light, it activates, opens this channel, and then leads to I think majority of the ions should be sodium channel entry to the cell membrane, and which cause the depolarization of the plasma membrane.
Okay, perfect. So the light active channel CHR2, and then it's open the channel, and the potassium and ions come inside and stimulate. Okay, perfect. Yeah, I'm true.
Okay, thank you.
This is in vitro system, but has anyone done this in vivo in mice?
Actually, we have done applied this technique, this method in the perfused pancreas, and it worked. So we can use this to stimulate. I'm not sure, but we can inhibit glucagon secretion when we applied light to darker cells.
Okay, great. Okay, I think, did someone else have another question? Feel free to unmute yourself. Okay, so I guess I would say, what's next steps for the laboratory? It looks, are you kind of commuting between Gothenburg and England? How is that working? How is this collaboration going?
Well, I am currently based in Oxford, but as a large part of our work is here in Gothenburg, so it does mean a lot of commuting, not as much as Professor Orshman is here every other week, but it means a bunch of commuting. But it turns out that a lot of researchers do that. I just found out, actually. It's not so uncommon to be in two labs at the same time.
I think it's great. I mean, it adds, it gives two separate environments, and probably feeds your, inquiry in different ways.
Absolutely, yes.
Yeah, more collaboration, the better.
The more collaboration, the better. Absolutely.
Yeah. And so, what's next for? Do you have a plan that you can share in terms of next sets of experiments, or is that kind of under wraps?
So right now, our optogenetic work is we work very close to being it's on the wraps, as you say, and we're doing some gene expression studies on the gap junction story, because it seems to be more and more evident that there's something there that we want to look into. Optogenetics has been a great tool for us, and so we have it feels like the possibilities right now, there's so many, there's so much we want to do with it, and also going deeper into both gene expression studies, but even some in vivo stuff, as well. Not translational yet. We're pretty much a basic research laboratory. So our work is our in vivo work is all in mice.
Yeah.
Yeah.
Well, right off the top, you can sort of think about, Coxsackie V virus and how that might be interacting with CX36, right? And that would be
What did you say? I didn't catch you. What did you say?
Coxsackie B virus, the CVB
Yes,
virus,
yes.
and how that might impact, interact with that CX36, component of the gap junction, and be so fun to like take some optogenetics and explore that, how it may change things. That's sort of like I've read some of that literature and it's very interesting. But is the Roseman lab looking for postdocs? This is the last question. And other trainees, are there projects to be had? Here's a chance to pitch that out to the wider audience.
So in Oxford, people join the labs mostly when they bring their own grants. So they are sort of like myself right now. I am a SSMF fellow, which is the Swedish Academy for Medical Research. And so we joined the group bringing our own funding and under the mentorship of the very gifted researchers there. Gothenburg, however, is a little bit different, but we are starting to have some really gifted people here as well that come with their own grants. So this is mainly the mode of joining the lab right now.
Okay, that's good to know. So if you're in the US or Canada or elsewhere and you just got a grant and you're interested in these kinds of studies, you can reach out.
Yes, and also collaboration. It's something that we're very keen on doing. We absolutely love working with other groups and having visitors here and being visitors elsewhere ourselves. So just reach out if you know something about optogenetics or just reach out. Do not hesitate to reach out.
Fantastic. Thank you both for sharing this really interesting work. Fantastic, dissection of what's going on here, and really appreciate your time and look forward to seeing what comes from your collaborations next. Have a great rest of your day.
Thank you very much. Thank you for this opportunity.
Thank you.